Okay, so today we're going to be doing our lecture 3, and this is therapeutics and vaccines. So we'll get started with that. And we're going to start off with, we're going to talk about DNA therapeutics, and then we're going to talk about protein therapeutics, and really we'll just focus on vaccines for that. So these are things that you can inject into a person, and hopefully they get better. And so the first thing we're going to cover is antisense. And so, you know, you have sense, which would be like the mRNA, and then you have antisense, which is going to be the complementary strand to that, the reverse complement of that. And it's easy to make antisense because you just can put the gene in backwards, right? So you just take the gene and you flip it over, and then with respect to the promoter, and you'll get an RNA antisense out of that. So let's see how antisense works. And so we look at this first one here. That's kind of small there. But basically the idea here, maybe we go back and get a better picture here. Basically the idea here is that you have a DNA, and then we have it transcribed, and we have a antisense oligonucleotide, and that binds to the mRNA of whatever gene you have in there. Or it could be an mRNA of a virus, for example. So one of the companies that's very heavy into this is ISIS, I-S-I-S, and it's in Carlsbad, California. And they did antisense. They started doing antisense work a long time ago, actually in the 90s. And what you do is you inject into the patient an antisense against a particular virus or against a cancer, a gene that might be promoting cancer, or some other gene that you want to knock down. And so the mRNA is bound by the RNA, by the antisense drug that you've injected into the patient, and then translation is inhibited because if you bind this especially to the AUG, or if you bind it to, oh no, okay. Well, we have yet another problem here. Okay. So let's go back and get started again. Okay. Oh, I see what it's doing. I know what it's doing. It was I pressed that one button and it couldn't display that, so it shut everything down. Out of courtesy. A free service. So if I don't press view original image, we'll be okay. So what it does is it binds that, and it can't be translated because it might be covering the AUG, for example, or the beginning of the untranslated region, the 5' untranslated region. So the ribosomes cannot actually construct, cannot actually land and begin translation. Now, antisense, I did a lot of antisense work in the 90s also, and we designed a lot of different antisense constructs that would fit onto different genes and tested them using in vitro translation. And it is true that doing it at the beginning, at the AUG, is effective, but some antisense molecules work fine farther down. Okay, since antisense came out in the 90s, a lot of things happened since then, and well, before I go on, there are two ways that this can occur. One is by stopping translation this way. The other is by RNase H. And let me go ahead and modify our terms here. So translation inhibition, we can go ahead and see what we get there. So it's steric hindrance, really. And so you can see this picture here, we just have the ribosomes are not able to bind, and so that's one of the things that happen there. You know, these are all little graphics that all look pretty much the same. But there are other ways that this can happen, and that would be by RNase H. And what happens here is that you have the DNA, you have the mRNA here, the antisense oligo binds to it, and then this attracts RNase H, and RNase H actually cleaves wherever the mRNA is bound by the antisense oligo. Now, as you recall, RNase H is will destroy. Here we go. This is good. RNase H will destroy double-stranded RNA, which is bound to another RNA, or RNA which is bound to DNA. So double-stranded version of RNA. It leaves all the others alone. And we have this RNase H built into us. And so if you bind a RNA to another RNA, to your mRNA, this will be regarded as foreign, and it will destroy it automatically. So which one is more predominant? Interestingly, it kind of depends on the antisense oligo you use. So in some cases, if you use one antisense oligo, it's RNase H which binds. In other cases, use another antisense oligo, it's predominantly translation inhibition, steric inhibition. So it just kind of varies. So since the 1990s, a big thing came up, and this was RNAi. And so with that system, we have, as you can see right here, you have a viral genome coming in, and this could be double-stranded RNA, right? If it's an RNA virus, it has to make another copy of itself. So let's say you're a plus-strand RNA virus, you have to make the minus-strand copy, so you can make more plus strands. So for a certain period of time, you're going to be double-stranded. There are no other double-stranded RNAs in your genome, I mean in your cells, only if you have a virus infection. So we have a primitive immune system which recognizes the presence of double-stranded RNA as foreign, as viral. Plants have this, insects have this, nematodes have this, and humans have this to some extent as well. And so this is a system which will recognize these, and it will destroy any double-stranded RNA. So that's RNA silencing. So what happens is, and we'll see this in just a moment, and I'm sure you're already familiar with it, we have the production of these silencing-inducing RNAs, these siRNAs. And they are, the silencing-inducing RNAs are recognized by, are picked up by RISC, and we'll see that in a moment. It's a complex for RNA silencing. And that has an endonuclease in it, and it cleaves the mRNA that way, at the double-stranded, well, not the double-stranded region. It cleaves just any mRNA, because it uses the siRNA that is grabbed here to identify the offending mRNA. And we'll see, again, we'll go over that in more detail. So when siRNAs came out, it just became the big thing, and antisense kind of went by the wayside, because siRNAs seemed so exciting. And everyone was into siRNAs, but interestingly now biotech companies are backing away from siRNAs. They are not working quite the way they thought they were. And now the biggest portfolios for ISIS are the antisense. We're going back to the 90s. So I think we're doing that also in music now, aren't we? Kind of going a little bit, using some of those retro stuff now. Anyway, so it's funny how technology comes and goes, but that seems to be, in the past couple of years, that seems to be the trend. So it's important to learn all the different technologies. You never know what's going to be ascending. So let's go over to siRNAs and to RNA silencing. Let's type in RNAi. Oops, that won't work. That won't work either. There we go. And so let's see if we can get some pretty pictures here for that. This one looks pretty good. Yeah. Okay, so what we have here is we have a double-stranded RNA, and that should not be in the cell. So dicer, or and this would be in nematodes, in other organisms, it's DCL, dicer-like, a dicer-like protein, will come and recognize that and cleave it into these siRNAs, which are about 22 bases long. And then this will combine, Argonaut is one of the proteins that comes in. We form the RISC complex around by binding to this siRNA, and it will go from double-stranded to single-stranded, and we form the RISC complex here, and it will identify target mRNAs using this particular single-stranded siRNA as the bait, as the model, as the template. And then it will cut that up, and that's the end of it. Okay, so that's that is RNAi, and that's a lot of money got put into that the past 10 years, but kind of pulling back a little bit. Okay, let's look next at another way to try to control RNAs. And so this is a wonderful goal because if you were able to shut down any mRNA you want, then you can do amazing things. As a matter of fact, before we go into this, I'll just go really briefly into just C. elegans and RNAi. And the systems they have for this, let's see if I can have this here. Yeah, well basically what you can do here is you can take nematodes and add double-stranded RNA and inject them into there, and you can degrade the target mRNA, and then you look for a loss-of-function phenotype. They're very amenable to that. But there are other ways that you can do it as well, and let me see if I can get this for you. Nice picture for you. You can actually have I'll tell you what I'm going to do. I'm going to put a vector in here. Maybe this will. Yeah, okay. So we have a vector here, and what you have then is you put your PCR product right into this slot here. And you'll notice there are two promoters, and they're going in opposite directions. So you're going to make the sense and the antisense version of the RNA corresponding to that gene. And so if you put that in E. coli, then you're going to get a sense and an antisense RNA being produced in the same E. coli. They're going to come together, and then you're going to have double-stranded RNA in the E. coli. And it just so happens that nematodes, C. elegans' favorite food is E. coli. They love them. And with the nematodes, you are what you eat. And so they have only 1,000 cells. And when they eat these E. coli, they ingest the double-stranded RNA, and this goes right into their cells, all over the nematode. It goes straight in. It's just amazing. They're so small. And so few cells. And now this RNAi can have its effect on the nematode. So you can actually just do your cloning experiment, get the E. coli, feed it to the nematodes, and turn off genes. It's really extraordinary. So it's a wonderful little system. Now that doesn't have to do with biomedical studies or anything, but it just shows you how powerful RNAi can be, and so why people's hopes were so high for it. And there's still some good research, good applications for it, but for biomedical, but that's where we were looking at. Okay, let's look for another one, and this would be ribozymes. And for here, what we have is a ribozyme is going to be an RNA which has some sort of catalytic activity. And in this case, we're looking for catalytic activity to destroy other RNAs. So here we can have a ribozyme right here, and then we have, as long as we have this one site that we can shoot for right here, this GUN, we can degrade, everything else can be any sequence that you want, as long as we have this one somewhere close by. If that's preserved, then you can have cleavage occurring, and the target RNA will be destroyed. So in theory, you can inject this into a person, and it will seek out the target RNA, and it seeks out the target RNA because this portion of the mRNA is not catalytic. It's just the part which recognizes the target sequence. This always has to be preserved, this bottom portion. But the portion which binds to the mRNA target, you can change to whatever you want. So you can have this recognize whatever you want it to recognize. And actually, Dr. Kane, I know Charles, you're working with Dr. Kane, he does this sort of thing, but with DNA rather than RNA. But these ribozymes have been used for quite a while. Okay, let's look a little more deeply in how these things work. Okay. Yeah, here's another example. So here we have the actual structure of the ribosome, and then as long as we have this GU, and then N can be anything, N stands for anything, and then right after the GU and then the N, we have a cleavage site. So the substrate can be anything you want it to be as long as we have a GU right here. And then the ribosome portion is this portion right here. So it's separate. Now let's look at ribozymes and look at how they are in nature. So we can look at ribozymes, for example, in virusoids. Let's see. Or actually, you know what we can do is we can do ribozymes in rolling circle. That's what I really want to do. And see how this would work. So here, a lot of, and these are viroids that we're looking at right now. Viroids are very, very small RNA pathogens. They're not even pathogens, we call them subpathogens in a way. Well, actually, these are pathogens in their own right. They do cause disease. But they're only 360 bases long, if you can believe that. So if you want the smallest bit of life, I guess you would call it this, only 360 bases long. Single-stranded RNA. The way that they replicate is they form this circle right here, and then once they form this circle, they start their RNA polymerase on one side of the circle, and they go round and round and around the circle, and they generate something that almost looks like a transcript, right? You're generating RNA from RNA. And it's very long. It's longer than the genome of the original viroid. And this is called rolling circle replication. You just go around and around and around the genome, and you make this concatemer of the genome. It's just many, many copies of the genome, head to tail. But then when you finish doing that, you have to have some way to break it down into the genome-sized pieces again. Well, this thing has only 360 bases. It can't code for any protein. It doesn't code for any proteins at all. It actually lives off of the DNA-dependent RNA polymerase, the RNA polymerase II, of the plant cell for it to replicate. So it's already borrowing another protein, hijacking another protein for its replication. What does it do to make this cut? Rather than coding for an RNase, which is specific for a particular site, it has built into the RNA a ribosome. And the ribosome occurs once per genome. So that once you have this concatemer forming, then you can go ahead and cut it. It will cut itself out automatically. And from what I heard, actually through the grapevine, people who were dealing with these sorts of RNAs, these sorts of plant RNAs, they would extract the RNA, and then it would be multimeric length, several different multimers of lengths. And then they would put it in the refrigerator overnight, and so they could analyze it the next morning, and it would have turned to monomers. And they said, gosh, what's going on? There must be an RNase in this. But a strange sort of RNase, because it would always cut perfect monomers. And so this was kind of a puzzle until people finally figured out it codes for its own ribosome. Okay? So that's what it is in nature, and what you can do in real life then is to go ahead and you can go ahead and use these then to attack other RNAs. And so, and of course some people felt that this is a fantastic technology, but anyway, so you can do this and it will actually code it. Let me get some other pictures on how this is done. There's a transesterification effect here. Here it is, right here. So what actually happens is there is the base, the cytosine base will catalyze this reaction, whereby you have a phosphodiester bond here between this nucleotide and the next nucleotide in the sequence, and a cytidine from another portion of the RNA, a cytosine from another portion of the RNA will catalyze this so that the phosphate binds to its own sugar right here. And now this is cut, and the phosphate is binding to the 3' and the 2' carbon of the ribose here, and then this is out of action. So that's how the cleavage actually works. All right, so that is ribozymes, and next we're going to go to aptamers as another version of something that will cut. So Celex is a method of doing this, of making aptamers. And what aptamers are is they are, let's see if we have a good picture here. Here we go. Yeah, that's good. This is the one I was looking at last night. The way this works is that you are trying, your purpose of this is you are trying to create a ribozyme. Or at least you're trying to create an RNA which can bind perfectly to a particular, I hate to use the word epitope, but almost an epitope, to a particular motif. So what you're doing is you're creating antibodies, in a way, that are not based on protein, but which are based on RNA. And the way you do it is you do an evolutionary approach of selection to find an RNA sequence which binds to a particular site. So you could have it bind to, for example, an HIV protein, glycoprotein, so that you can have a diagnostic used to detect HIV. Or you could have it bind to a protein which is commonly found in cancer cells, expressed on like a TAA, a tumor antigen, and tumor-associated antigen. And you could have it bind to that one, and maybe you could disable cancer that way. Or you could use it as a diagnostic. So as a therapy or diagnostic, it's just like creating antibodies, except you're creating RNAs. So how do you do that? How do you make an aptamer? The way you do it is you have a random region in the center. Just start off with randomness. And then you have a constant region, perhaps here, which might be better defined. Then you have a T7 promoter, and you go ahead and make transcripts. Well, if you make transcripts, in this case, we're making 10 to the 33rd different sequences. Now, obviously, you can't make quite that many sequences because you might not have that many molecules, but you make a lot of different sequences because we have a random region right here. And then you take those sequences and you go ahead and expose them. Here are all the different sequences. We give them different shapes here. And then here's the target. It's a particular protein. And you run this through a column containing your target. So let's say that you have Rev, let's say, from HIV, and you want to target Rev for some reason. So you have that protein which has been expressed, has been produced. You bind it to a column, and then you run all these RNAs through there. And you go ahead and wash them. And you wash out all the ones that don't bind. And then the ones that do bind, at least somewhat, you are retained, and then you elute those out, and these are RNAs, right? So we go ahead and do reverse transcription on these RNAs. Then we have the cDNA. We go ahead and do a PCR amplification on that cDNA. Because remember, we have only a very few molecules that have bound. With the PCR amplification, then, we have a whole lot of DNA of different types, because there are different types that bind to this. And then we go right back to where we started. We then go ahead and do a transcription, and we make the RNA again. And now we go ahead and bind it to a fresh batch of Rev protein sitting on a column. And hopefully these will bind a little bit better. And then we go ahead and wash, but this time our washing is going to be a little more stringent. We might vary the salt concentration or the pH or something like this. And now we're going to get rid of more of the RNAs, and we're going to go ahead and elute this cream of the crop that comes out, and then we repeat it again. We reverse transcribe, To recover what bound, we PCR amplify to get it to a good level. Then we do the transcription again, and we bind again to fresh target. And this time we wash even more stringently. And you keep on increasing the stringency of the washing and do it over and over and over again, maybe a dozen times or more. And then finally what you wind up with is RNA, which binds so well, these particular sequences bind so well that it's very hard, they stick to it very well, it's very hard to get any of them to wash off. They stick quite well. And at that point, hopefully you have a diagnostic product or a therapeutic product. And those are called aptamers, and the CLEX procedure is the company name for that procedure, for the protocol. Okay, I'm going to stop this video and then restart another one so that we have smaller videos at least to deal with. So.